2, 1. And we're live.
We'll also give some minutes to allow people to enter into the room.
Sure. Oh, what the heavens happened there?
Okay, I think there is people that have entered, and I just wanted to welcome everyone, and we'll get started in a few minutes. Just want to give some extra time for people to enter, and then we'll get started. Okay, I think we can get started now. Good afternoon, everyone, and welcome to Youth Neuro Australia's 2023 Brain Awareness Week webinar. My name is Emilia, and I will be your host tonight. So, just as a side note, please note that today's webinar will be recorded. To begin, I would like to begin today by acknowledging the traditional custodians of the land on which we stand and pay my respects to their elders past and present. I extend this respect to Aboriginal and Torres Strait Islander people here today. For those of you who don't know, Youth Neuro Australia is a youth-run non-for-profit that focuses on providing STEM events, resources and education to high school students and university students, focusing particularly on neuroscience. If you'd like to know more and get involved or hear about our upcoming events, feel free to follow us on social media and chat to one of our volunteers to get connected. So Brain Awareness Week is all about demystifying brain research and making it accessible for everyone. So today's webinar will be focusing on the gut-brain axis, and Youth Neuro Australia is very excited to be joined by an expert in the field, distinguished laureate Professor Nicholas Talley. Professor Talley studied medicine at the University of New South Wales. He graduated in 1979 with honours. He undertook graduate training in internal medicine, becoming a gastroenterologist. Interested in research, Talley received a National Health and Medical Research Council scholarship in 1984. In 2018, Dr. Talley was awarded the New South Wales Scientists of the Year Award, as well as the Peter Mills Medal. Dr. Talley is currently Pro-Vice Chancellor of Global Research at the University of Newcastle, and has made discoveries leading the forefront of gastroenterological research, particularly neurogastroenterology and associated disorders. So today, Dr. Talley is going to talk about his research and why it's important to us, all of us, regardless of your science background. At the end of the presentation, there will be time for questions where you can ask anything, including questions about Dr. Talley's presentation, research, clinical work, or anything else you're curious about. So if you think of any question, make sure to put them in the chat function on the side, and just type it in there, and we'll announce the questions to Dr. Talley to answer. So without further ado, you can take it away, Professor Talley.
All right. Well, thank you very much. I hope you can hear me okay, and I hope I'll make this a bit of fun. I'll cover some science, but I'm really going to try to make this at a very, very high level and talk about gut-brain, because the gut talks to the brain, about microbes, because microbes talk to the brain through the gut and elsewhere, about diet, because diet changes the microbes that changes the talk to the brain, and about health and disease, because everybody cares about health and disease. So I hope it'll be fun. I hope you'll enjoy it. I hope you'll learn something, and I hope it'll stimulate you, maybe even change your perception of who you are. So let's go with that. I'm also going to acknowledge country. I'd like to begin by acknowledging this meeting where I'm standing is being held on the traditional lands of the Awabakal peoples, and pay my respects to them and elders past and present, and extend that to Aboriginal and Torres Strait Islander peoples viewing today. Now, some of you may have seen this show. I don't know, The Last of Us. I've been watching it myself, actually. It's actually a really good show. And it's what most of people think about microbes. Microbes are the enemy. That's what people think. And in this show, a really interesting show, a fungus has taken over the brain or the brains of many people in the world, and they turn out to be zombies. And the actual fungus taking over the brain is actually a fungus that exists in Brazil. And this fungus takes over the ant brain and controls ants. And that is actually true. So this fungus really does do this. It just doesn't do it in people. So this is how many of you will think about microbes. They're the bad guys and we need to do something about them. But actually, microbes are our friends, often. And if you look at the movie War of the Worlds, which was written a long time ago, the book was written a long time ago, and the movie's much more recent, microbes in this story, they're our saviors, because we, as humans and all animals, we co-evolved with the microbes so that we can live with them in health and harmony. And we're resistant to disease these microbes would otherwise cause. But if you've never been in contact with these microbes, they can and they will kill. And in fact, in history, this happened with diseases that were introduced into, for example, the Americas by the settlers there 300, 400 years ago, when they brought smallpox and other diseases which devastated the indigenous populations of those parts of the world, because the people had never been exposed to the microbes causing those diseases. So microbes can be our enemies and they can be our friends. And in this story, by the way, the microbes end up killing all the alien invaders, because they'd never seen those microbes ever before they invaded the Earth, at least in the story. So microbes are absolutely everywhere. And not only are they out there, they're inside all of us. We are made up of microbes as well as human cells and human components to our bodies. The organisms living on and in us make up about 1 to 3% of the body's total mass. So that's a couple of kilograms of bugs that actually live on us and in us. And these are bacteria, these are fungi, these are viruses, and there are also organisms called archaea. And these are the main groups of organisms that live in and on us. And these organisms, they're very, very important for our health, but they're also important in disease. So they can be both friend and foe. So you all know what bacteria are, but I'll just remind you, bacteria are tiny, microscopic, single-cell organisms. We can see them under a standard microscope. They're found absolutely everywhere on Earth, even in the coldest places, even in the hottest places, even in the deepest places. They are everywhere. And some of them can live in extremely severe conditions of temperature and pressure. They're amazing, amazing organisms. Archaea are like bacteria, but they are single-cell organisms that don't have a cell nuclei. And some of these organisms, the reason they're important actually, is they modulate our health. For example, archaea can produce gas in the body, gases in the body, in the intestinal tract. And one of those gases is methane gas. And methane gas actually slows down the movement of the gut. And in fact, too much methane gas can lead to constipation. So really, this is a group of organisms that are there. We don't know that much about them actually, but they can have health effects. Fungi are eukaryotic, nucleus-containing organisms too, and these include yeasts and moles, and of course out there growing in the garden there can be mushrooms. They're also fungi. They differ from bacteria because of their cell wall composition, and they generally decompose organic matter. But they also are present in our guts and elsewhere in the body, on the body, and they have different roles there in, again, health and disease. And then perhaps the most interesting microbes are the viruses, because viruses are barely alive. Some people would argue they're not alive, actually. Certainly they're at the edge of life. They infect everything living. Us, other microbes, they infect everything. They can be DNA or RNA containing, most are RNA viruses. They can cause disease. We actually don't know very much about what they're doing in our bodies most of the time. There's a big area of research going on looking at viruses in and on us. So when you look at all these microbes, if you add them all up, there are 30 trillion human cells in the average human. That's you and I. And there are 39 trillion microbial cells on average. In other words, you and I are only part human, whether you like it or not. You are not totally human. In fact, you could argue there are more microbial bits of you than there are non-microbial bits of you. And if you look at the genes, we have about 20,000 human genes in the cells of human cells, and there are, well, somewhere between 2 and 20 million microbial genes in the average human. So we are about 43% human in terms of cells and about 1% human in terms of genes. So this is an enormously important active part of our bodies in every single human that's alive today. If you had no microbes at all, if you were born with no microbes and acquired no microbes, you would not survive. You would die. Microbes are part of keeping us alive. They're part of our healthy constituents of our bodies, and they interact with our bodies, but they're also separately alive and have their own interests, which is replicating and feeding, and we'll come back to that. So most of them live in the gut, in the intestinal tract. We'll come back to that, and they can be altered by diet, by exercise, any drugs you take or medications, antibiotics, and early life exposures. So very important. So when I say microbiome, I mean in a human, all of the microbes, that's viruses, fungi, archaea, bacteria, and all of their genetic material in the body. That's what the term microbiome means. And it's a whole habitat, as I've said, a whole ecological habitat that co-evolved with us over hundreds of thousands of years. The metagenome means a collection of genes from the microbes. It's just a term. And the metabolome means what the microbes are making, the chemicals and other products they're making, which, by the way, are very important for our health. So why all the interest in this in recent years? And there's lots of reasons, but the main reason is in the past, we could not grow most of the organisms living inside of us. Even though we tried, we couldn't grow them in normal growth media that we can grow other bacteria and things in. But what's happened is we've developed techniques to look at DNA and RNA as well, techniques that will tell us actually what is there. What are the organisms that are there? We can also measure what they do, what's called metagenomics, or what they could do, what their genes would allow them to do, and then what they actually do, what's called metabolomics, metatranscriptomics, what they produce, the metabolic products and the RNA products that change things as well. So the microbiome's very important. It's mostly in the gut, but it's in your mouth, on your skin, in the vagina. It's everywhere on your body. You cannot get clean of these organisms. In fact, you wouldn't want to, because that would be really unhealthy. Everybody is unique, though. Every single one of us has a slightly different combination of organisms. So we're all a little bit unique. And that's also important to recognize. And the amount of data, sort of storage here is just remarkable. If you look at how much bacterial DNA there is, and if you wanted to sort of measure it, you'd have to have a lot of thumb drives, old-fashioned technology, I know, but still worth thinking about, a lot of thumb drives, 100,000 or so, to be able to actually get all the data from 1 teaspoon of stool, of bowel movement material. In the diagram here, this is a statistical technique for separating out humans from the environmental organisms, the bacteria, for example, that are present. The humans are in green and the environments, the other colours, and humans look different. The bacteria, for example, in humans are different to the bacteria in the environment. Again, we co-evolved. We are somewhat unique. All animals also have microbiomes just like this, although they are different again from human microbiomes. In the gut, most of the organisms live in the large bowel. That's the place where we store our stool before we're ready to evacuate, before we're ready to get rid of it, which all of us have to do, every human has to do to remain healthy. But there are organisms right through the gastrointestinal tract. In the swallowing tube, the esophagus, there are some in the stomach even, even in the presence of gastric acid, which is normal in the stomach, and gastric acid's really toxic, there are quite a few bacteria present there. In the small intestine that connects the stomach with the large intestine, there are also reasonable numbers of bacteria, more than the stomach, but most live in the large bowel. There are also lots of organisms in your mouth and in everybody's mouth, lots and lots of organisms that are important for dental health in this situation. So the gut microbiome plays a very key role. Why is it so important? Because from early life, from the moment we're born, we acquire organisms from our mothers first, typically. If we're born vaginally through a normal way, then we will acquire the mother's organisms. If we're born with cesarean section, where they cut the baby out, then actually you first acquire skin organisms and other environmental organisms before you acquire your mother's organisms. So you get a different microbiome if you're born by vaginal delivery versus cesarean section. The organisms live in us and they shape our immune system. Our immune system notices the organisms, makes antibodies to them often, and actually they talk to each other. They talk to each other, which allows the immune system to mature successfully and protect us from other organisms that are much nastier pathogens that can kill us or make us very, very ill. The bugs also help our lining of the gut to be solid and not leaky. A leaky intestine can cause disease, and the presence of organisms help it to not be leaky. So terribly important. And the organisms in our gut, they harvest some of the nutrients we need to survive. Some of the nutrients we must have only come from bacteria interacting with food. Our bodies can't do it. The organisms are needed to make it happen. So isn't that interesting? We've co-evolved to live in harmony, hopefully, when we're healthy, with the organisms in our body. But of course, the harmony can be overturned. We can have alterations here. And if we have disharmony, if things go wrong, we get what's called dysbiosis. If you like, disturbed microbiome presence in our bodies. It's a little bit unclear what's really healthy and what's really diseased, though. There's a bit of a spectrum. We know some things are bad, but we also, in healthy people, sometimes see those bad organisms and yet they remain very healthy. So lots of research still needed to work out what exactly is healthy and what exactly is disease. And every disease is going to be a little bit different, which makes it complex, but absolutely fascinating. You can collect your stool if you want, your bowel movements, and you can send it off to a lab to get your microbiome tested. And let me tell you, don't bother. Don't bother, because we don't really know, except in some cases, what it means when you get the results. That's why you do research. And when you do research, you've got to control for lots of factors that can alter these microbes. And there's lots of issues around this. When you collect the samples, what you're eating when you're collecting the samples, or recently eating when you collect the samples, how you store and ship the samples, how you test the samples, all of that can change the results of an analysis of the bugs in your bowel movements. And that makes research a little bit complex, but what you do is you control for those factors and you make sure you have healthy people and diseased people, and you compare them, controlling for the factors like diet and other things that can change the results and mislead you. That's science, that's research, and that's what we do in my group and elsewhere, many groups around the world. So what do we do then? Well, It gets complex, and I don't want to go into this in detail. But what we do, for example, is we can pick a certain part of the gene that all bacteria have, and there's a sequence of that called 16S, and just about all bacteria have that sequence. And we can look to see, if we test that sequence, what bacteria are actually present, because we can put together the way the sequence is determined. And in fact, we know that for lots of bacteria, and we can map it to, if you like, a computer, or using a computer and using some other things to be able to work out exactly what's there. But if we really want to know, the best way is what's called shotgun metagenomics, where we break up the DNA into very small segments, and then because of multiple overlapping reads, we actually, after several rounds, can put them back together to again work out what is actually there in terms of the genetics, and then map that to the organisms that we know about, and also find novel organisms that don't map to anything, but are similar in some ways to other organisms we know about, so we can work out what genus and species they are. I know that sounds complicated, and I'm sorry about that. I'm just saying here we can do this, and this is a picture sort of how you do this really. It's about doing the DNA extractions. It's about shaking it up with and splitting it, and then looking and reading this against various reference databases and coming up with what's there, and also what the genes might be doing can be calculated. And finally, we can look at the metabolic products present as well, and we can try to work out which bacteria, for example, are producing those metabolic products. So very complicated, but that's what this is all about. And these novel high throughput technologies have just absolutely transformed how we think about and what we know about these bugs that live in and on us. And that's been incredibly exciting. So what have we learned? So we've learned, for example, that when you're unborn, basically you are sterile. You don't really have any bugs inside your mother's womb. At least we don't believe so, or not very many. But once you're born, you very rapidly, within hours, acquire microbiomes in the skin, in the mouth, in the gut, and elsewhere. And hopefully you then grow up to be a healthy child and then healthy adult. And as you age, your microbiome ages with you. It changes. And when you're very elderly, you have a different distribution, for example, of certain bacterial species that's quite different from when you were a child. And some people believe that may be a clue to working out how to reverse aging. And I'll come back to that, because wouldn't that be exciting, although we're a long way from being able to do it. We also know, as I said, diet's very important, and so you need to control for that. That's something that will change the microbiome as well, and obviously your diet's different from childhood to when you're an older person. So there are things there that make it complex, but even adjusting, controlling for diet, you can see very substantial differences between the elderly and younger people. And so this is really important, and we know healthy bacteria are associated with better brain function, better thinking, better memory, and less inflammation. And inflammation is what causes many, many, many diseases that we know today as chronic diseases that we see in usually adulthood, although sometimes, unfortunately, at younger ages. So what diseases are linked to changes or alterations in the microbiome? And there's a whole list of them. Inflammatory bowel diseases, asthma, brain diseases or neurological diseases like multiple sclerosis, diabetes and obesity, lots of cancers, Alzheimer's disease and other neurodegenerative processes, depression, anxiety, and even other mental syndromes, and then of course irritable bowel syndrome, which I'll talk about briefly later on. So lots of different chronic diseases are linked to abnormal or dysbiotic microbiomes in the gut. So we're talking about gut microbiome and disease. It doesn't mean the microbiome is the cause of the disease. Got to be very careful here. It could mean that, and in some cases it almost certainly does. But it's also possible if you get a disease, you may change your diet and your lifestyle so your microbiome changes then too. So in fact, the microbiome is not causing the disease, it's secondary to having the disease. So think about that. It's not always cause and effect if you see an association. It may be the other way around, what's called reverse causality. So how does the microbiome talk to the brain? Why do you get neurodegenerative diseases in this situation? And the answer is the microbiome has a critical role, as I mentioned, in the immune system, but it also produces products and interacts in other ways with the nervous system in the gut. And the gut is sometimes called the second brain of the body. There are lots and lots of nerve cells in the gut, more than anywhere else except the brain. And they talk to the brain through the vagus nerve and other connections. And those nerves are incredibly important in digesting food and making us feel very comfortable after we eat, and also in allowing us to pass bowel motions comfortably and normally. So that's really important. So what about the immune system? Well, here's a cartoon about this, and I don't want to go into great detail, except to say in health, the normal side of this figure, the bacteria are there in the gut lumen, in the lining, just above the lining of the gut. There are cells there, what are called antigen-presenting cells, that put their noses through the lining of the gut. You can see the purple cell doing that. And they're there to sense if there's an invader or a change. And if they sense this, they'll set off the immune system. But in health, this doesn't happen. The immune system isn't firing abnormally. It's just a normal balance, a homeostatic balance. But if you get inflammation for some reason, for example, you get an infection and that leads to the gut becoming inflamed, and you get a leaky gut, then bacteria can come through the lining. The antigen-presenting cells sense the bacteria, sense they're in an abnormal place, and the immune system attacks. It fires off. It fires off B cells, which produce antibodies, and T cells, which can kill organisms, and also memory cells, T cells, which can come back and remember this going forward. And you may get a cycle where the inflammation continues because the gut has overreacted to the initial insult. And so this is how diseases can develop and occur. And we think this is very important in lots of chronic diseases, from brain diseases to other problems around the body. We also know that undigested food's available to gut bacteria. Gut bacteria eat the food in some cases. They'll produce things, chemicals like short-chain fatty acids, and these short-chain fatty acids can then actually talk to the nerves and change the nervous system. And that goes back up to the brain, and the brain may react in a way that changes behavior, but also leads to conditions like abdominal pain or other problems that people commonly have as they are going through life, which we know about. And there's lots of this metabolic stuff that's transformed by bacteria, lots and lots, from the diet, like amino acids or sugars, polyphenols, from what we produce in our bodies, bile, for example, or cholesterol or steroid hormones, which we all produce. That's just normal. Bacteria can change these chemically and lead to metabolites, to signals that can again interact with the nervous system. And then There can also be other biosynthetic precursors that can lead to toxin production even, that can also potentially lead to signalling to the brain. So really a complex system. It's not very simple, but the point is it's incredibly important, and it's happening every day in health. But things are good in health, everything's under control in health, but in disease, things go awry, they're out of control. And the immune system plays a key role in this, as I've said. So lots of things change the microbiome for good or for worse. For worse, caesarean section birth, well, we can't change that if we were born that way. Lots of antibiotics. Antibiotics are a disaster. The microbiome, the bacteria in the microbiome, get markedly changed by a course of antibiotics. It can take months and months for it to recover. It may never recover after a course of antibiotics. So overuse of antibiotics is incredibly problematic, particularly in childhood. A very clean environment is actually bad, not good. Very clean is actually unhealthy. You need to be in contact with microbes for health. Isn't that interesting? Too clean, it's not good. Having no contact with animals as you grow up, again, we need that contact. Some of the organisms we need, some of the organisms that help us come from other animals. And so that's also important. I've mentioned diet already. High anxiety or stress, that can change the microbiome for worse. So obviously it could be a vicious cycle. You get anxious, you change your microbiome, and you get more anxious. Or it could be your microbiome gets changed, then you get anxious, and you get into that vicious cycle. It could be both ways. So there you go. And there are some things that are better than that, and I've mentioned those on the other side of the slide. So what can we control in our own bodies to stay healthy? Well, we can control what we eat. We can control exercising or doing exercise. We can control what we put into our body, like supplements or drugs and medications, and we can control, to some extent, sleep. And we need all these things: good sleep, maybe not supplements necessarily, but sometimes certainly exercise, and definitely a good diet to stay healthy for life. If you eat badly, your microbiome gets sick. So eating McDonald's, for example, all day and all night is not recommended. It's okay to have it sometimes, but not recommended, because in fact that's an unhealthy diet, and it will alter your microbiome significantly if you live on a very high fat, very high sugar sort of diet. Not a good diet at all. Low fiber too, even worse. When we eat, what happens is we mix it up in our mouth. Not much happens there. There's a little bit of breakdown of things, but not very much. In our stomach, there's acid, but it doesn't break things down. It just basically mulches it together, and then it all gets absorbed in the small intestine. When it gets to the large intestine, right at the bottom, that's where the poo is. When it gets there, that's where the fermenting, feeding bacteria live, but they're also incredibly important for our health. So actually those bacteria that live in that large bowel, all of those bugs, they are there to help us. And we have a lot of bugs in the poo that we get rid of, but there's an enormous number of bugs living in that large bowel too. And what's undigested in the top part, in the small intestine, gets down to the large bowel, the colon, and in that area, bacteria will break things down. Some of that we use for nutrients. Much of it, though, the bacteria use for themselves. But again, those bacteria there are important for keeping us well. So eating lots of fiber, for example, helps to keep our bugs healthy, helps to keep our colon working well, helps to avoid developing constipation later in life, helps all of us to remain generally healthy. And none of us eat enough fiber. Most of us in Australia and around the world, in developed countries, don't eat enough fiber. We don't get anywhere near the needed 40 grams of fiber a day. One of the best diets, if you ask what's a really good diet, it's called the Mediterranean diet. And it's high fiber, it's full of fish. It's also got eggs, interestingly. So eggs are healthy, actually, even though they're high fat. It's got nuts and lean meats, so not very fatty meats, because that's where there can be problems. And you avoid or limit salt and sweets. And that diet is actually a very healthy diet, one of the healthiest diets in the world. And people who live on that diet tend to live longer, and possibly because their microbiome is healthier over time. And then there are other crazy diets, the gluten-free diets. And all I can tell you about gluten-free diets are very unhealthy unless you've got celiac disease, when you must be on a gluten-free diet. A gluten-free diet is high fat and high sugar, because it has to be, because you move out the wheat products. And actually that changes your microbiome too, and not necessarily for the better. So these fad diets, many of them are just really unhealthy, and your microbiomes don't like them. So remember that. So I'm a neurogastroenterologist. I work with lots of disorders of the gut, and some of those disorders, perhaps most of them, are linked to alterations of the microbiome. So let me talk briefly about one of them, very briefly, and this is the irritable bowel syndrome. And why do I talk about this? Because this is a disorder generally of younger people, people under 50, often in their 20s and 30s. That's when they develop this disorder. You can develop it as a child. You get abdominal pain and bowel disturbance and bloating. There's no inflammation that you can find, but we do know the microbiome's altered in people with this disorder. And we think that's relevant to the gut-brain connection that we feel is so relevant to this disorder and many disorders that affect health, including gastrointestinal health. These are just the symptoms that people have with irritable bowel syndrome, the classic symptoms, and their bowel habit, the way their poo looks, is also abnormal, and that's associated with the pain people report with the syndrome. We know psychological distress, anxiety, depression, multiple other symptoms, they're all linked to IBS. And one of the questions has been, could it be the microbiome not only drives the development of IBS symptoms or keeps them going, could it be the microbiome and its connections to the brain? Could it be it accounts for community or for the psychological distress that people very often have with IBS? It's very, very common. And we know your gut talks to your brain. I mean, I've already said this, but you can actually show it using what's called functional MRI. That's measuring the blood flow in the brain. And what we do in this experiment is you blow a balloon up inside the rectum, inside the lower end of the large bowel of people with IBS versus controls. And the brain response is different in IBS versus healthy controls. Their microbiomes are also different. And so very interesting work. And certainly the brain and the gut talk to each other. We know that. And this is just one piece of evidence that's the case. So what we try to do in some work we've done is trying to work out what comes first. Is it the psychological distress that comes first and then IBS? Or is it IBS comes first and then you develop psychological distress? So we've done some work on this, for example, some work here in Newcastle, Australia, where we've assessed people in the community with IBS. Remember, nearly 10% of people have IBS. It's really, really a common disorder. If you haven't got IBS yet, you might just get it. So there you go. It's an important, common problem in Australia and around the world. And what we found in this research is about 1/3 of people first develop anxiety problems and then they get IBS. It looks as if the brain talks to the gut and that's what sets off the problem. But 2/3 of people, 2 in 3, It's the other way round. The problem begins in the gut. There's a disorder of the gut becomes abnormal, and then later on they develop anxiety, maybe depression and other features. In other words, the gut drives the brain. And why is that important? Because we can potentially address gut problems much more easily than we can address brain problems in terms of interventions. So that's really important. So what may cause IBS in the gut to the brain? Well, we think the microbiome plays a role, but we know it interacts with food, it interacts with things you do like smoke, and it also interacts with infections that are common in the gut. So I'll just talk about that really briefly. And this is the role of the microbiome in irritable bowel syndrome. And I'm going to talk a little bit about this. I'm not going to go through this diagram here, which is very complicated, but it's basically based on the evidence that's been accumulated about the changes in the gut, including in the immune system and in the nerves that can occur in this syndrome. So we know, for example, that if you put certain foods or you eat certain foods and they go from the stomach into the small intestine, which is they always do, that's normal, we know you can actually develop an acute inflammation if you have IBS. About 50% or more will develop an acute inflammation from foods being exposed to the lining of the upper small intestine. And that's very exciting. And we think what happens here is the bacteria interact with the food. They lead to the foods releasing antigens or allowing antigens to present to those immune cells that are waiting to sort of taste them, and that leads to an acute inflammation that causes the symptoms people feel, and it goes away rapidly too. So it comes, it goes, it comes, it goes, which is what IBS is like. It comes and goes. So what can you do about this? Well, you can go on a diet, and for example, a low fermentable carbohydrate diet, that's what a FODMAP diet is one way to take out a number of substances that can potentially cause the gut to become either inflamed or can lead to other alterations of the microbes that can be relevant to the signaling of disease. And this diet works in about 70% with IBS, about 70% who go on a low FODMAP diet or low fermentable carbohydrate diet, they will improve substantially if they have irritable bowel syndrome. And this also changes the microbiome. What else can you do? Well, you can try probiotics. These are live organisms that you eat. So these probiotics contain live organisms, billions of them, in a little pill, for example. And some probiotic strains may be helpful, although many probably do very little because they don't last in the gut. They just go through. They actually, the other microbes that are present, they block these microbes, these probiotic microbes, from attaching. So in fact, you don't get much response at all. And then what you can do as well, at least theoretically, is give a poo transplant. Who's heard of poo transplants? Huh? Well, poo transplants date back to Chinese medicine in the 4th century. Yep, 4th century. That's where it began. And in fact, in recent years, in the last 10 or so years, there's been enormous interest in doing this to change the microbiome, not only of irritable bowel syndrome, but of inflammatory bowel disease, and also if you get bad infections of the bowel, which can respond very well occasionally. A certain specific infection called Clostridium difficile can respond to a poo transplant, or the medical term is fecal microbial transfer. That sounds much nicer than poo. How do you do it? Basically, you homogenize the poo of a donor and you can place it into the large bowel, or you can get people to even swallow it, although not like this, I can assure you, as a pill, for example, or if you put a tube down into the stomach, you can put this material through a tube. So that's how you do it. But the donor you use needs to be healthy, and you need to make sure they haven't got any other serious diseases that you could potentially transmit from giving poo from one person to another. So that's the complexity of doing this. And so if we do this, and it's only really used in a few indications now, if we do this, we need to make absolutely sure the donor we select is coming from a donor bank that's done all the screening, or that the screening has been done by us and done properly, so that the safety is maximal, and that's incredibly important. But for IBS, just to show you some data, to show you that I'm not talking through my hat, you can see if you give a fecal microbial transfer to IBS patients in a randomized control trial, that means you randomize people to placebo, dummy preparation, no poo, to FMT 30 grams, that's a sort of low dose, or FMT 60 grams, that's kind of a higher dose. Well, you get a response, a significant response above placebo giving the poo transplant. People got better. That's what that means. And this effect lasted in this study for 3 years, which is remarkable. The problem is we don't know exactly what's in the poo that makes a difference. That's really important to recognize. And we don't know if it's safe for 30 years to give poo. Remember, we might transmit a virus by mistake, for example, that we don't screen for, that we don't know about, that might cause another kind of disease. So you only do this kind of work for IBS in a sort of research study. It's not part of standard medical care at this time, or it shouldn't be at this time. And in mice, we can actually reverse aging in mice. Now, mice are not humans. Mice are not. So you've got to be very careful when you extrapolate. But giving a healthy fecal microbial transfer to mice, actually from young mice to doing this to age mice, actually improved their health. And so really interesting that indeed it might be possible in the future to improve health by using techniques like fecal microbial transfer, or hopefully a better approach would be to identify what's in the poo exactly that makes the difference, and giving that substance or substances rather than everything in the poo, which would be safer and certainly more palatable. So microbiome is really important. It's changing our whole view of the world we live in, and the world of disease and health. And in the future, we're going to see huge work in this and many advances that will hopefully improve health over time. Maybe even prevent cancers, maybe even prevent Alzheimer's, maybe even reverse, partially at least, aging. All theoretical, but certainly relevant. So feed your microbes, folks, feed your microbes. The better you eat, the healthier you will be lifelong. Eat plenty of fibre. Don't use antibiotics unless you absolutely have to. Keep your stress levels down if you can. Exercise regularly. Your microbes will love you and you'll stay healthier. You may even prevent nasty diseases that otherwise might have affected you if you do this. And then in terms of the science, in the last minute or two, what we're doing here and many other labs now is we're doing something called culturomics. What we're doing is we're actually looking at those DNA signals, and then we're trying to grow the organisms that we've identified but have never previously been grown. That's what we're doing, as are many others. We're studying what used to be unculturable microbes, and we're having really exciting results. Here's an example. We found a bug in the upper small intestine that's linked to a syndrome a bit like irritable bowel syndrome. And we did that by doing, first of all, the metagenomics, doing 16S in this case, and that's what that bar graph shows, and then putting the biopsy tissues, inoculating them, growing them, and then finding bacteria on the plate and then identifying what they are using whole genome sequencing and matching them to databases. And by doing this, we found 3 novel streptococci, which is a common type of bacteria. And the streptococci, one of them in particular, seems to potentially be one of the, we think, potential causes of the gut syndrome we've been studying. And this is just one example of what will be happening down the track as we find causes, we hope, of disease that we can treat in the future, perhaps even cure. So it's very exciting. That's the kind of research we do. Here's sort of a sort of summary of some of the work we're into, from celiac disease to Crohn's disease to IBS to other gut disorders. And it's just fun, to be perfectly frank, and we love doing it. And of course, this work doesn't happen alone. It happens in a big group with lots of great people and lots of great collaborations. And it's very exciting to work with other scientists to learn, to explore, to find new things that others have never found, and hopefully to make a difference to health as well. Thank you very much. Happy to take any questions.
I'm not sure I understand.
Yilka, you're muted.
My apologies. I'll start that again. Thank you so much, Professor Talley, for your talk. It was incredibly thorough and informative. We really, really appreciate it. So on behalf of everyone, thank you. We do have some time for questions. So I know some people have already put some through in the chat. Feel free to keep doing that, and also feel free to vote for any questions that you think you'd like answered as well. So we might start with, if you could please talk a little bit about what neurogastroenterology entails clinically day to day.
Okay, so it's a subspecialty of gastroenterology. So as a gastroenterologist, we see patients who have gut diseases or liver diseases. That can be inflammatory bowel diseases, it can be bowel cancer, it can be celiac disease, it can be ulcer disease, it can be all sorts, reflux disease, all sorts of diseases that are commonly affecting the gut. But there's a group of both common disorders and very rare disorders where the nerves and the muscles of the gut are particularly abnormal. We think they are not functioning correctly. So irritable bowel syndromes are a really common one of these. Like, as I said, nearly 10% of Australians, wow. But there are rare diseases like what's called gastroparesis, where the stomach fails, or very severe constipation, where the large bowel fails, doesn't work properly anymore. That's neurogastroenterology. So it's good fun. And the microbiome's incredibly relevant to almost all of the diseases we're talking about. So that's the reason I've ended up in the microbiome as a research area.
Awesome, thank you. One quite popular question is, what is your opinion on the widespread use of prebiotics by the general public for quote unquote overall gut health?
So the answer is we do not know what we're doing exactly. Some prebiotics are probably quite helpful for health, but we don't really quite know what's the best approach. The best evidence for health is the Mediterranean diet, which is a combination of different things that you do. There's not a lot of evidence that prebiotics really improve people's health. And there's a lot of stuff you can buy at the chemist, in the supermarket and elsewhere, which is probably utter rubbish. No offense to those who make it, you know, and because there's just no evidence that they work. A lot of the products they sell over the counter, really, there's not much science behind many of them. And that limits, I think, what we know. So, yeah, I don't know what else to say. There's so many products you could go, you know, ad nauseum through a whole bunch of them. But I think the bottom line is, most of the things we buy for health at the chemist, you know, supplements, vitamins, all of that is probably for most people, not everybody, but most people, absolutely of no value. Basically, you're spending your money and losing it. Eat
It's a very
a good
clear
diet.
answer.
Eat a good diet. That is cheap, and that's a good idea. That's a really good idea.
Excellent. Thank you. Another popular question is, when did you realize, a bit of a different question, but when did you realize the massive impact of your clinical examination textbook? So this person has said, speaking on behalf of most, if not all med students out there, it is our Bible. But did you think it would be this influential when you were working on it?
So the answer is absolutely no. I did not think it would be influential. I remember writing the textbook. So I wrote it after an exam that I'd sat and finally passed, and that exam led me and my colleague Simon O'Connor to write a textbook, and then 2 textbooks and a few others. When we wrote, I remember when we wrote it when we wrote this book, and I realise a lot of people don't read books anymore; they study online, etc., etc. Although these things are online too, of course. When we first wrote it, I honestly thought if 10 people bought it, that would be the best thing that could ever happen. Like, that would be fantastic. I'd be amazed that 10 people would buy it. And I really wrote it for me. I wrote it because I wanted to communicate there were better ways to teach what we were being taught. And this was, sort of not out there. No one had really done it. So I've been flabbergasted that people actually still like it. Obviously, we've done new editions since the first edition, but regardless, it's remarkable. So thank you to the question. I don't know what else to say, except I'm humbled that people still find it valuable, and that's great.
I think we have a little follow on from that as well. Another person said, I really enjoy reading your clinical textbook. Will you consider creating another clinical examination textbook in the future, maybe in a simplified version?
Well,
we can quickly refer to.
That's a great question. We did do a simple version. There is one out there, sort of an introductory version, so a much shorter, smaller book, which you can buy. It's not as popular, I think, because probably the big book covers it all, and the small book's more a summary kind of guide. And we thought for introductory medical students that'd be helpful. And it's still in print. We still do new editions, but it doesn't sell in the same way, and I can understand that. So that is available. But I don't know. I think the more you know, the more you learn, the more you read, the better you'll be. So, reading the bigger textbook and perhaps getting as much information into your heads as possible can be very useful when you're training to be a doctor, in my view.
Yeah, thank you. So back to maybe more of a scientific question. How can we overcome the issue of correlation versus causation with the gut microbiota in systemic illnesses? Just a small question for you there.
It's a fantastic question. So much of the work is what we call association. So people with a disease have a different sort of group of, for example, bacteria in different species and genuses versus controls. So that's kind of association. That does not mean the bacteria are doing anything in terms of health. Or disease. It just tells you they're different when you measure them. And the differences could be because people are eating different diets. So the diseased people might be eating differently to the controls. They might have different exercise levels. They might have other factors that are different. So you've got to sort of adjust for those. But the most effective way to work it out is to intervene. The way to intervene is to find the organism that looks as if it's the best candidate for potentially being a disease driver, and then showing that you can actually put that organism into an animal and cause disease and treat the animal, and the disease will go away. So that's one way. Or to treat the organism in a human and show it improves disease in a randomized trial. And it's got to be a randomized trial because you need to have a control group that's very similar to the group that you've given the intervention to. So a randomized trial of diseased people, one group gets whatever it is, the targeted therapy, and the other group gets a placebo. So there's clearly you can show a difference. So it's the intervention experiments, it's the old Cox postulate approach that eventually leads to the most convincing evidence of real cause and effect. And a lot of the data at the moment is association, and of course some of it's wrong, and we just don't know which parts are wrong yet.
Beautiful, thank you. So there's a question here. Is there much known about the clinical/immunological effect of formula given to babies compared to breastfeeding mediated by the microbiome?
Yeah, look, this is not an area I know a lot about. It's not an area I study. There are differences between formula-fed and breastfed milk. Breast milk oligosaccharides, which are sugars in breast milk, they have positive effects on healthy microbiomes. In fact, we're studying breast milk oligosaccharides to see whether they improve brain function and gut function. And I'm very excited about this, because if they happen to work, that would be really, really, a very nice way to intervene, because they're very safe and they're available. So that would be a sort of prebiotic story that might work. So they are different, and they do affect the microbiome. Breast milk remains, as far as we understand it, the best way to feed babies, if that's the choice you have, because of not only the milk, but also the bacteria that the mum gives through doing breastfeeding.
Awesome, thank you. Do we have time for one more? Is that all right?
Yeah, one more sounds great.
Beautiful. I was one of my questions is, how generalizable are these results across, say, different cultures or different socioeconomic groups, because of the way that environment has an impact on the microbiota?
There's no doubt there are differences across countries and groups, and you've got to think about that as you do work. Makes it a little hard sometimes to compare diseased groups in one place versus diseased groups in another. And sometimes they look very different, and we're not really sure why, whether that's related to other factors we don't know about, or whether in fact the genetics play a role, because genes, our own genes, also interact with the microbiome and affect how the microbiome looks in an individual. So there's genetic issues as well. So it's really complicated. And so gene, environment, microbe interactions is where a lot of work is being sort of looked at the present time. But you're absolutely right. There are clear differences, and you need to consider those whenever you're looking at results, and also when you're designing experiments trying to test what's going on.
Awesome. Thank you so much. We'll have to leave it there today just based on time. So sorry for everyone who hasn't had their questions answered. But on behalf of everyone, thank you so much for talking to us today. We really appreciate all the knowledge you've imparted. And as has been evidenced by the questions, you're quite an inspiration to a lot of medical students out there. So thank you.
Well, thanks all for listening, and it's been a great pleasure. Thank you.
Thank you. Just for our audience, we do have a feedback form that's been posted within the Q&A as well. So if you would be willing to fill that out, that would be awesome. And also don't forget to follow Youth Neuro Australia on our socials, or even apply to get involved so that we can set up more events like this. But thank you everyone for coming, and we hope you have a great night.